Okay, and so in this video, we'll talk about how gene therapy is used or how it could be used in the future. And, the ethical concerns that show up and even the new technology called CRISPR that you may have heard about in the news. All right, so, you know, what's gene therapy? Basically taking this recombinant DNA technology and these molecular tools that we're learning to change the genes of individuals. Imagine someone born with a genetic disease of just a protein that does not work. Okay, let's put it simple, like the simplest terms. If they are born with the protein that does not work, and you are able in the womb to take that fertilized egg out, put in a protein on that zygote, and put it back in the womb, implant it, and have that baby grow and have that working protein, that is the ultimate goal of gene therapy, right? And that's what you see over there, right? Our pro can solve a debilitating illness just like that, just by adding a protein. We do it in plants all the time. It works. It's called complementation. Okay? We can knock out a gene on purpose and then put in a working function of the gene. And if that plant now works normally, we complemented it. It's what the ultimate gene therapy would be. Okay, so we know it could work, but it's where do you draw the line, right? And so that's where we get into some of our cons, right? We could insert our genes unintentionally into our eyeball genes because we don't tell it where to go. Okay, we could unintentionally use a modified virus and it reverts back to being infectious because it got infectious to begin with, right? So that happens somehow. Could happen to you. Um, we also get into the fact that it may not actually help and it may end up damaging by maybe making a constitutive promoter that runs too long. Lots of things could happen. Okay, so the ultimate goal is to help a one gene, one illness problem. Okay, that's the simplest terms. But we can get into some ethical concerns here again, like we want to always kind of keep in mind, especially going into healthcare, what genetic traits are worthy of being corrected, right? Is it just a one gene, one illness? What defines illness, right? Um, that's not for me to decide. It's just for me to raise that this actually draws a concern. This is the big one, right? Should the gene therapy used for cosmetic reasons? Oh, I want uh, lighter hair. I want non-disposition to, uh, you know, uh, thyroid problems, or want to not have male pattern baldness in my 20s. Where do we draw the line? Okay. Um, do we do that to the unborn, or can we do that to alive people? It's a lot harder to do it in a live person because we'd have to do every single cell in our body. Much easier to start in the zygote stage where the cell just is going to divide naturally. And if we can put it in that cell, every cell will divide just like we learned about in mitosis. And then, are we just introducing more social inequality, right? Can everybody in another location than the United States, can they afford it? Can everybody in the United States afford this, right? Or will they just have to realize that, they don't have this protein and that's it, right? Whereas other people can just do it just because they made a lot of money. And then finally, who actually regulates this? Is it the UN, right? Is it the US, right? Is it the state? Definitely lots to think about when you think about gene therapy. But it's really cool to think about in terms of plants and insects and microbes, and how we can use these tools to study what genes are needed, what genes of metabolism work, and use them as tools to study rather than as tools to design and impart these ethical concerns on anybody. To further the knowledge is not to raise the concern, right? The concern comes from what you do with the knowledge. All right, and with that, we talked about off-site targeting. That's a coined term, off-site targeting. That means, is the gene going to land in a gene that's already working, right? Because of randomness, that's where the bacterium is just landing, right? So when that happens, that's called off-site targeting. You go somewhere you don't want it to, right? So, but CRISPR here, CRISPR takes advantage of a bacterial mechanism that defends itself against viruses. So a viral infection will induce a prophage, right, into the bacterial genome. That prophage can be located by these by this mechanism's proteins, and it will locate that, and then a protein comes and will cut each strand of DNA twice, once at each rung of the ladder at two different places. Those cuts out uh, the infectious strand, the prophage, at least at little points. Again, if we just do a little bit of deletion, we're going to offset frame shift and we'll just destroy the protein. We don't need to get rid of it all. So we just destroy the harmful part, and the harmful part's usually the conserved part, so it's easy to target it, right? And we cut it out, and then our natural machinery will rejoin it, DNA ligase. Okay, so this is a useful tool. I've used it in the lab. It's really fun. You do definitely reduce off-site targeting. You can even put in the whole genome, and it can tell you where it would target. And you can say, oh, that's acceptable. That's in this set of DNA. That's acceptable. Or you say, oh, time to change it. Okay. And so with that, we have the end of our DNA tools lecture. And I hope you enjoyed a small dive into the world of molecular biology.